Well, just now I saw some students asked about the attendance, and I understand because the technical problem, and for recent several times, the DingTalk is not available for everyone, so we switched to this dual app, dual platform, and some of them attend the lecture in Ring classroom. That's okay. Don't worry about your attendance. Well, because of this DingTalk problem, we need to rethink about how to calculate the attendance. Maybe we'll just use the data before the crash of DingTalk, at least for the evening group. For the other group, we are okay. We can use the DingTalk data. Okay, so let's continue for the human immunodeficiency virus. That's short for HIV, human immunodeficiency virus. This is another pandemic we are experiencing in recent 30 or 40 years. So though it's not obvious, but it's a real pandemic. It's influencing all the world, everyone in the world. Maybe you think that HIV is far away from you. You're not influenced, but who knows? A lot of things can happen. A lot of accidents can happen. Later, we'll talk about the virus itself, and we'll also talk about the related story about HIV. And okay, let's go. HIV can cause a disease called the acquired immunodeficiency syndrome, short for AIDS. That's famous. The virus first come to people's attention in early 1980s. In big cities in America, such as Los Angeles and also New York. At that time, the first symptoms of the viral infection is not a viral infection, because at the beginning, people even didn't know there is some virus infection. They discovered some cancer on the skin or in the blood muscle. That cancer is called Kaposi's cancer, which is very rare, but it got popular somehow in those big cities. And another interesting property of that is that disease got popular only in those gay societies. So it even got the name of gay cancer. And for a couple of years, people didn't know the reason of that. And people guessed maybe there is some infectious agent there. But they didn't know whether it's some bacteria or some other virus. And they tried to figure it out, and labs in both France and America, they tried to discover the real cause of that disease. And after a couple of years, they independently discovered the virus. Well, according to some gossip, the major contribution is from a French lab. And then later they shared the Nobel Prize by discovering HIV. And they now realize that It's not gay cancer. It's just some virus infection, and the virus is infectious and can be transmitted. And former names of HIV can be, in other words, if you read a very old literature, you will keep in mind that this is the same thing as HIV, such as a human T-cell lymphotrophic virus, because HIV prefers to infect T cells, or lymphadenopathy-associated virus, or AIDS-associated retrovirus, ARV. But these different names all refers to HIV. Here is some characteristics of the virus. Structure of the virus is icosahedral symmetry. There is envelope outside the virus, so it's enveloped. It's lentivirus, which means that this virus, after infection, the replication of the virus is pretty slow. Subfamily of retrovirus. We learned retrovirus before. It's RNA virus, but after infection in the host cell, this RNA genome can be reverse transcribed into DNA, and the DNA fragment can be inserted into the host genome. So this is a special virus, retrovirus. And of the virus, there are some important proteins on the envelope, especially the GP120 and GP41, which are involved in viral infection. And they are recognized by receptors on the host cells. Later, I will show you how they interact with host cell receptors. This is some characteristics of HIV. This is how they look like under electron microscope. And you can see the envelope, making them spherical-like. And intracellular, this is the nucleocapsid. It's a icosahedral structure. This is the genomic organization of HIV-1. Right now, there are different strains of HIV. The majority of patients are infected by HIV-1. You don't have to remember those structures, but keep in mind that there are important proteins in the genome, including some structural protein, some enzymes, some coat proteins, and all together they make up the viral particle, the virion. There are three main genes. First, the group-specific antigen, short for GAG, group-specific antigen. That's the structural protein. And on the envelope, there are two glycoproteins. One is GP120, the other one is GP41. And they can make polymerase. This polymerase is a reverse transcriptase containing P66 and P51 subunits. Together, they make the reverse transcriptase, which can make DNA from RNA. Okay? So remember these important proteins: GAG, group-specific antigen, the envelope protein, and also polymerase. Okay, here is the important glycoproteins, including GP120 of HIV. GP120 is this one, the blue one. It protrudes from the outer envelope and responsible for virus attachment to the CD4 molecule on the host cell. You know, CD4 is the marker of helper T cell. So that is why HIV infection prefers the existence of CD4. And there are some co-receptors, including CCR5. We'll talk about that later. And GP120 can also elicit neutralizing antibodies. But even though GP120 can elicit neutralizing antibodies, there are some diversity, and the mutation of the gene is very quick. So it's not easy to make vaccine using GP120. Well, GP41 is a transmembrane glycoprotein, the red one. It can help the virus to anchor on the host cell and facilitate the viral penetration. Later, I will show you how the viral infect the host cell. Here is the interaction between the viral and its receptors. This is the viral particle, HIV particle. It expresses both GP41 and GP120, and the GP120 first recognized by CD4 receptor. So this is a CD4 molecule, the purple one. And also it can be recognized by cytokine receptors. That's the co-receptor. After binding to this CD4 T cell, well, usually it's CD4 T cell, but some other cells can also express CD4 molecule, for example, the macrophages. So HIV can also infect macrophages. This co-receptor here is CCR5 or CXCR4, which are important. In some populations, for example, in the Caucasians in Europe, the white people, they have some natural mutation in CCR5. There is a fragment of the gene get deleted. So that's a variant of CCR5 gene. And that receptor is not functional. So that means that co-receptor for HIV is not functional. So that makes the HIV infection very poor. So some people in carrying that gene are kind of more resistant to HIV infection. This is the HIV life cycle. I think previously we introduced this life cycle before. So first attachment, the viral attached to the surface. And because it's enveloped, it contains a membrane. There is a membrane fusion, the second step. And it can release the viral nuclear capsid into the cell. And after uncoating, it releases the genome, the viral RNA, the viral RNA into the cytoplasm. And there, the reverse transcriptase can make DNA from the RNA molecule. First, single-stranded DNA, then double-stranded DNA. This double-stranded DNA is transported into the nucleus, and there, it's integrated or inserted into the host cell DNA. This makes it provirus. And from there, it can transcribe to make mRNA. This mRNA can be translated into proteins to make viral particles. And also, the mRNA can be reverse transcribed to make protein to recycle. And this mRNA is also their genome and can make new viral particles. So this is a life cycle. This is CD4 positive cell, host cell. Comparing the size of HIV virus on the T lymphocytes, you can see the big difference. All the background is a T cell. It's part of the T cell. And these small dots, small particles are the HIV virus. So comparing to the host cell, the viral particles are too small. They are just look like ashes on the surface. So they are very small. Pathogenesis and immunity. Pathogenesis and immunity of HIV. The sources of transmission. Usually, patients with HIV or patients of AIDS or some asymptomatic virus positive individuals. That means they are infected, but they didn't show any symptom, and they can release the virus to other people, other individuals. The transmission route of HIV infection can be in different ways. The first route is the sexual contact. That happens a lot. Maybe it's a homosexual, a heterosexual, and it can transmit virus. And for the beginning in the early 1980s, the homosexual transmission is a major type because those people in that community, they don't have the pressure of a pregnancy, so they are not protected. So they got exposed to HIV infection. Another type of infection is the blood transfusion or blood products. So that's usually that's not a problem because either blood transfusion or blood products are well controlled. They need to test it carefully, make sure that it doesn't contain any virus. But the problem is that even though you detected the existence of known virus, but if there is some unknown virus, you cannot find it. And this happens for HIV, and this also happens for HBV, because before people realize there is a virus called HBV, they they cannot test it in the blood, so they use that blood for transfusion, and that caused some problem. Okay, and for HIV, that's a sad story. The blood transfusion caused a lot of infection in China, like about 30 years ago. That's a good that's an example of blood transfusion. You know, 30 years ago, that's when I was a little kid. It's a long time ago. Most of the people are were already or still very poor. They cannot find jobs. They cannot work. So finally, they find a way to earn some money. That's to sell their blood. There are some companies collecting blood to make blood products to earn more money. So at that time, some people, especially in the poor villages, they sold their blood to get some money. But the procedure is not carried out perfectly, and it caused transfusion or it caused infection of HIV. Maybe there was some cases, some individuals positive, but finally it infected a lot of people here in China, especially in our province, in Henan province here, and got some infection. So that's a sad story. But for now, the blood transfusion is not a big threat anymore. Another way is the contaminated needles. This usually doesn't happen in modern hospitals, but it can happen in some societies. It's, for example, the drug abusers, because they use drugs a lot. They spend a lot of money on it. They don't have time. They don't have money to buy a new syringe, a new needle. So they share the syringe, they share the needle, and that can cause a lot of transmission. Or there might be some vertical transmission. So that's from infected mom to the new baby. For this transmission now, they can the hospital can help a little bit. They can help to isolate the sperm and isolate the egg from both the father and the mother, and then make the fertilized eggs and ask another person to have their baby. So that can be expensive, but it's a way to have their own baby. This is the course of HIV infection. That's onset of the disease. Let's talk about these lines. First, the purple line. The purple line is the number of CD4 positive T cells, the trend of the T cell count. While for the green line, that's the HIV RNA copies in the plasma. So that's the, well, you can think that the green line is the HIV load. So that's the RNA copies in the plasma in each meal. Okay, so this is a green line scale, the y-axis. You can see this is the exposure time. Time 0 is exposure time. And within a short time, this is weeks, within a couple of weeks, the HIV can be detected and increases dramatically in the blood. You can detect the RNA directly in the blood. It's onset of the infection. But quickly, the immune system is turned on, and it can control the virus infection. And you can see there's a big drop now. So this is the first stage of infection, acute HIV syndrome. And during this primary infection, the CD4 positive T cells will decrease dramatically. So the virus can infect T cells and clean out the T cells. But after that, T cells will come back a little bit. This becomes the clinical latency period. This latency period can be several years, can be 8 or 9 years. That's a long time. So during this time, usually the infected patient doesn't show any symptom. They look healthy at this period. Or even though their T cell number is low, it's lower than normal, but they look healthy most of the time. But sometimes they can have some co-infection. For example, like bacterial co-infection, especially MTB. So that's tuberculosis bacterial infection. And after about 8 or 9 years, the clinical latency, there will be the onset of the disease again. This is the final stage of the disease. The patient's T cell will decrease again, get lower and lower, and the viral load get higher again. So that means the immune system is too weak now. It cannot handle HIV infection anymore. So HIV load get higher again. Then after about 10, 11 years, the patient will come to the death. So this is a natural infection of HIV without any treatment, this course. Good news is that right now we can give the patient some antiviral medicine. We can control HIV infection. Well, for some extent. You cannot cure HIV infection, but you can keep the patient live alive, relatively normal. So this is the description of that figure. I don't want to go over it again. It's technically it's what we talked about just now. Symptoms of HIV. For acute infection, are non-specific and it can include like fatigue. The patient will feel tired, rash, headache, nausea, and night sweats. So that's acute infection. The patient is characterized by a pronounced suppression of the immune system and can develop a wide variety of severe opportunistic infections, including like the MTB infection. And some patients will develop unusual cancer, especially the Kaposi sarcoma. And with no treatment, finally, after 8 to 10 years or 11 years, the patient will die. So technically, this is what we covered just now. And this is Kaposi sarcoma. It's a rare cancer of the blood vessels that is associated with HIV and can be diagnosed on the skin. And you can see these bluish red oral shaped patches, and these patches can be single or can be clusters. This is the Kaposi sarcoma. Co-infection. We mentioned that MTB infection is one of the co-infection, that's bacterial infection, but the fungi infection can also happen. This is the fungi co-infection of Candida. So we call it the oral candidiasis in the throat. This is a fungi infection. For immunity, HIV-infected persons develop both humoral and cell-mediated responses against HIV. In most infected individuals make neutralizing antibodies against the virus, directed against the enveloped glycoprotein, that's GP120, as we mentioned just now. But the levels of neutralizing activity are low. Many anti-envelope antibodies are non-neutralizing. So those antibodies, most of them cannot work. Okay. The envelope glycoprotein shows great sequence variety. That's why we talked about just now. This may allow the evolution of a resistant virus that escape recognition by existing neutralizing antibodies. This, our immune system are not deaf. They are there. They can sense the HIV infection. They did respond, but they cannot handle it for finally. For laboratory test of HIV infection, we can isolate the virus. We can use the virus to infect cells. Or popularly we can use a serological test to detect the antiviral antibodies, or we can detect the viral antigens. Or we can use the viral nucleic acid to detect the HIV DNA or HIV RNA. It's an RNA virus, so you can detect the RNA in the serum. Because it's retrovirus, when it infects the host cell, it's integrated into the host genome. So you can also isolate the total DNA and detect whether there is viral DNA in it. Treatment. When we talk about the treatment of virus infection, previously we talked about HIV treatment a lot. Several slides were covering HIV treatment. You can design medicines against different targets in HIV. For example, you can design inhibitors to inhibit the reverse transcriptase so that they cannot make DNA from RNA. Yeah, that's correct. Or you can use some inhibitor against the protease to inhibit the maturation of viral proteins. So that's a protease inhibitor. But clinically, right now in hospitals, they use a cocktail. So cocktail means that it's a mixture. So in those medicine, it contains several antiviral medicines. They combine them together to suppress the viral replication. And to suppress the viral replication for a long time. So that's we call it for common people, we call it the cocktail treatment, cocktail therapy. After about 30 years, or after almost 40 years of discovery in HIV, right now we have developed several medicines, and with the help of the medicine, HIV patients or AIDS patients can live a relatively normal life now, and their lifespan is almost normal as healthy persons. So that's a good progression we made, but still we cannot cure them. We cannot cure HIV patients. We cannot cure AIDS patients. So that's the sad news. You may heard of the story about some person cured after they infected by HIV. Maybe I can tell you that story. There was a young man who was American, I think. He went to Europe and for either study or something, but he got infected by HIV. So that's not the worst news for him. He got HIV infection. He got treatment with a cocktail therapy, receiving medicine. And several years later, he got another disease, which is leukemia. So both HIV infection and also blood disease. What the doctors did is that to give the guy some treatment to make the blood transplant, well, the stem cell transplant. So they find some donor to donate stem cells for this guy. So to help him, they destroyed his immune system. They destroyed his own stem cells because it's a blood cancer. Yeah, it's a blood cancer. So they give this guy stem cells from another person to restore his blood system. And yeah, this guy recovered. And the doctor reset his blood system. And what's surprising is that they found later that this guy is HIV negative. They can never detect the HIV virus anymore. That's interesting because they didn't expect that. They tried to figure out the reason. They finally found that the donor stem cell contains a variant of CCR5. That's what we mentioned just now. CCR5 is the co-receptor. That's a co-receptor of HIV. And the donor cell, the stem cell from the donor, contains a mutant of CCR5, so that the HIV virus cannot infect the new CD4 positive T cells. So finally, this guy recovered, and he lived for a long time. I think he passed away recently, and that's the only person, as I know, that's the only person, or at least uh, one of several person cured HIV infection. But that's not for everyone. You cannot find the stem cell with that mutation for everyone to find that donor stem cell. For now, we don't have vaccines available against HIV. That's not good. And for most of the patients, most of the AIDS patients, we cannot cure them. We can help them to stay a normal life, but we cannot cure them. Recently, for this coronavirus, we designed mRNA vaccine, and it's available in America. Right now, over 2 million people have got mRNA vaccine, and it worked pretty well. And hopefully, with this new revolutionary technique, we can finally design some vaccine against HIV. And if HIV vaccine is available, that's a good news. Before the end of the lecture, I would like to introduce this lady to you guys. This is Dr. Gao Yaojie. Well, she actually graduated from our university. Well, it's not Zhengzhou University. She graduated from our medical school a long time ago. She graduated in 1950s. She's 90 years old, 91 years old now, 91 or 93 now. She graduated from our medical school a long time ago, 1950s. And she worked as a doctor in Zhengzhou City for all her life. Dr. Gao retired from a hospital. And after she retired, that was in 1990s, she realized that there is some disease in this province. That's the HIV infection, AIDS. But at that time, that was early. In China, no one believed there is AIDS patients in China. There is HIV infection. But Dr. Gao realized the problem and tried to investigate and try to find out the infection, try to find out the patients. And what she found was that there was a chain of blood trade, and the poor people are selling blood to get money, and the companies are collecting blood, and those people selling blood are not well protected. There is transmission. There's blood transmission. Yeah, it's some accidental event, but it's kind of popular. A lot of people got infected in the poor villages. Dr. Gao found out the problem, but yeah, at that time she also faced a lot of pressure from the society, from family members, from the government, from the companies. Yeah, and she faced a lot of pressures. But her work was recognized by international organizations and by other countries, and she got some prize, and now she is living in New York, in Manhattan, somewhere in Manhattan, all by herself. That's not a good story. Because of her excellent work, her excellent investigation on AIDS infection in China, her family broke up. and she divorced, and her children complained about her, and yeah, her family broke up, and she finally went to America. She is now living in New York. So that's a real doctor, actually. That's a real doctor who are really brave to figure out the problem and let everyone know and let everyone be careful about infectious disease. Yeah, she graduated from the same medical school you are starting now. You are alumni. So this can be your role model, Dr. Gao. Yeah, she did sacrifice her life. Uh, that's she retired before she began the work. In China, if you retired as a professor, as a doctor, you can live a very good life without any pressure. But she sacrificed a lot. This is Dr. Gao. Okay, here are some video for you guys. Do you want to watch here? Do you want to watch these videos? No. Okay. Actually, you can watch those videos on the Superstar platform. You can watch there. The origin of HIV is a long documentary video, and people are trying to figure out why the HIV virus spills over to human. Uh, we think that the source of HIV is from animal. Some animal in Africa, especially the chimpanzees. For some reason, it spills over to human and cause this pandemic. Okay, I think it's better to leave these videos for you guys. You can watch anyone on the Superstar platform. So okay, so that's all for this one, and I'd like to show you some example questions. Hold on, please. Okay, we have some example questions. Okay, can you see my new PPT showing the example questions? Okay, so the example question, I will just here show you some. Well, someone asked, well, how the virus got spills over from chimpanzees to humans. I understand your concern, but probably it's not a sexual contact. It might be some accident. Some hunters in Africa, they hunt the chimpanzees for meat, and when they kill the chimpanzees, maybe they got the virus by accident. So that's the possible way. Yeah, they ate raw meat, or when they killed the animal, the blood got contaminated their body, and somehow it spills over to those hunters. And yeah, about the time, the time point. I read a book on different viruses. The first cases were identified in America in 1980s, but the spillover might happen much earlier, maybe 100 years before that, from the animal to human. Okay, here are some example questions. We'll just start this one. Ignore this. The first type of questions are the multiple choice. For example, this one, most bacteria increase in number through the process of which one? Can anyone tell me? Can you still remember this type of this question? Bacteria increase their number through which process? Binary fission. Great. Yeah, you got a point. Yeah, I will send this file to you. Another one is, which hepatitis virus can spread viral fecal-oral route? Can you see? A, B, C, D. Which one? A. Yeah, exactly. HAV. And also HEV, right? We just talked about it. Which of the following is not true related to endotoxin? This is related to bacterial endotoxin. A. Endotoxins are secreted from cell or can be linked to meningosemia produced by gram-negative bacteria, can cause fever. Which one is not true? Yeah, exactly. A. A is not true. Endotoxins are not secreted. Okay. Which is not true. Okay, keep this in mind when you take your final exam. Okay, be careful. You need to understand the question. You need to pay attention to whether it's true or not true or not. Just pay attention to the question. A is not true. Endotoxins are not secreted by the cell. It's a structural component on the outer membrane. So it's not secreted. When the cell dies, when the gram-negative cell dies, it releases the endotoxin. It's not secreted. Okay? Okay, this one. ELISA technique can be divided into what types? Competitive ELISA, sandwich ELISA, indirect ELISA. D, yeah, shown here, D. All of this. ELISA can be carried out in different ways. Another type of question is this one. It's a true or false question. You just pick the answer whether the statement is true or false. First, flu viruses have two kinds of spikes on their envelope. They are HA and NA, hemagglutinin and neuraminidase, right? That's true. Sterilization is the total destruction of all organisms, all microorganisms, including most resistant forms. That's true, right? And the last one, differentiation between Enterobacteriaceae is based on gram stain. Is that right? That's not right. Yeah, the answer is wrong. So because Enterobacteriaceae, they look like to each other. So you cannot tell the difference only according to gram stain. Okay, so that's the example questions. So yeah, we are done for our lectures and we are done for our course. And thank you guys for your attendance for this past 2 months. And we had pretty good time together for these lectures.